I think we can start this session, sir. Am I audible to all?
Yes, yes, please
Yes, sir.
continue.
Okay, thank you. Good evening to all the distinguished guests, respected professors, fellow academicians, researchers, and dear participants. I, Pavan Pandey, Assistant Professor, Rajiv Academy for Pharmacy, Mathura. It is a true honor to welcome you to this faculty development program. On behalf of the Indian Pharmacy Graduate Association, Mathura Local Chapter, and Rajiv Academy for Pharmacy, Mathura, today we begin an exciting 6-day online national faculty development program on exploring innovation in pharmaceutical sciences for better future. We are here because we believe in sharing knowledge, growing together, and building a brighter future. In the days ahead, you will hear from amazing experts, discuss fresh ideas, and discover how the world of pharmacy is changing for the better. I wish to express sincere gratitude to Dr. Devendra Pathak, President of IPGA Mathura Local Chapter, and Professor Dr. Himanshu Chopra, Vice President of IPGA Mathura Local Chapter, Director of Rajiv Academy for Pharmacy. Their dedication, vision, and gentle leadership have played a vital role in scheduling this program, a great success, and in nurturing a culture of academic excellence. I heartfelt thank you to our respected chief guest, Dr. Atul Nasasar from SGT University, Gurgaon, and our guest of honor, Professor Dr. Arun Garg from MVN University, Palwal. Your presence makes this event truly special. I also want to appreciate our hardworking speakers, resource persons, and the organizing team for their dedication and support in bringing this program to life. To all our participants, I encourage you to join and ask questions and make the most of this opportunity to learn and connect. Let's work together to inspire new ideas and spark innovation in pharmaceutical sciences. Once again, a warm welcome to each of you. Let's make these 6 days full of learning, discovery, and success. Now, to formally, this is the time to formally inaugurate today's session. I would like to invite Dr. Mayank Kulsresht, Organizing Secretary and Professor, Rajiv Academy for Pharmacy, Mathura, for the inaugural speech. Sir, please.
Thank you, Mr. Pavan Pandey. Thanks a lot. A very warm good evening to all of you, all the chief guests, guest of honor, all the participants, respected chief guest, honorable guest of honor, esteemed partners, conveners, distinguished speakers, faculty members, research scholars, and all my dear teachers. It is needed to my great privilege and honor as the organizing secretary of this 6 days online National Faculty Development Program on the topic, which is basically exploring innovation in pharmaceutical science for better future. To welcome you all inaugural session. The pharmaceutical science today are at the crossroad of transformation with rapid advancement in drug discovery, formulation technology, regulatory science, and patient-centered healthcare. The role of academia and research has become more crucial than ever. This faculty development program has been thoughtfully designed to bring together competent academia research professionals from diverse domain of pharmaceutical science to discuss, deliberate, and share knowledge on the innovation that will shape the future of healthcare. Our object is not only to update faculty members with the latest trend in pharmaceutical science, but also to create a platform for the discussion, exchange of ideas, forecasting, collaboration between academia and industry. We believe that tough, such as academic endeavor, we can contribute meaningfully towards creating better therapeutic outcomes, promoting impactful research, and nurturing the next generation of pharmacists and scientists. On behalf of Organization Secretary, I extend my heartful gratitude to our chief guest, Shri Atul Nasaji, which is a Pro Vice Chancellor, SGT University Gurugram, and sparing valuable time to grace the occasion with an inaugural address. We are equally grateful to our guest of honor, Shri Arun Garg Sir, Vice Chancellor, MVN University, whose presence add great value to this program. Welcome, sir. I take the opportunity to sincerely thank our patron, Shri Ram Kishore Agrawal our chairman, convener, Professor Dr. Himanshu Chopra, our co-convener, Dr. Vivek Joshi, for their constant guidance and encouragement in making this FDP a reality. A special thanks also goes to our esteemed resources person who will enlighten us during the session over the coming days. But last, not the least, I warmly welcome to all participants who have joined us from different parts of our country. Your active involvement and feedback will be the sure strength and success of this FDP. I am confident that this 6 day journey will be intellectually stimulating, enriching, and will inspire all of us to explore new dimension of pharmaceutical science for a better future. Thank you once again, and I wish the program great success. I heartily thanks to I heartily welcome to all my chief guests, all my participants. Kindly join the FDP, ask the questions to the speakers and enhance your knowledge. Thank you. Jai Hind, Jai Bharat.
Thank you so much, sir, for setting the tone for this FDP with your inspiring words. Thank you, sir. Now, it is needed an honor to welcome our chief guest of the day, Dr. Atul Nasha. First of all, I would like to introduce our chief guest, Professor Dr. Atul Nasha is the president of Indian Pharmacy Graduates Association and managing trustee of its welfare trust, a distinguished pharmaceutical expert and academician. He currently serves as Vice Chancellor of SGD University, Gurugram, holding PhD in pharmaceutical sciences with over 25 years of in drug regulation. He has served as Deputy Drugs Controller at Delhi Drugs Control Department, honored with the Best Drugs Control Officer Award in 2017 and Fellowship of Indian Pharmaceutical Association. Dr. Nasa is an Executive Council member of Pharmacy Council of India, actively contributing to its law and other committees. He is a life member of several professional bodies, including IPGA, IPA, APTI, IHPA, AIDCOC, ISTE, IPS, Indian Society of Pharmacognosy, and Indian Society of Cosmetology, as well as member of International Pharmaceutical Federation. A noted resource person, he has conducted numerous training programs, seminars, and consumer awareness initiatives for NGOs, academic institutions, and government organizations, renowned for his visionary leadership. He has represented India in global forums, advancing pharmacy education, regulation, and research. Now, I would like to invite Dr. Nasa to kindly address the gathering. Please, sir.
Thank you very much. I'm audible.
Yes, sir. Yes, sir.
Thank you
Yes,
very much.
sir. Yes,
Thank you
sir.
very
Yes,
much.
sir. Yes, sir.
So, good evening to one and all. At the outset, my heartiest congratulations to Dr. Javinder Pathak and the faculty members of Rajiv Academy for Pharmacy that they have taken this initiative to have this 6 days faculty development program for pharmacy professionals. An important role in drug discovery, in drug repurposing, in personalized medicines, in gene therapy, in 3D printing, in clinical trials, in supply chain management, and so on. This is how artificial intelligence is playing a very vital role in pharmaceutical industry. We have seen that during the pandemic time, two vaccines like remdesivir and tocilizumab, basically they were not meant for treatment of COVID, but it was drug repurposing as these injections are used to increase the immunity in the patients when the immunity is down. So this remdesivir and tocilizumab played a very important role during pandemic time by increasing the immunity to the patients. My sincere request and advice to all the participants attending this webinar and faculty development program that there is a bright future for pharmacy in the different fields to try to guide all the PG students, all the PhD students that the future for pharmacy is very bright. As initially I told that lot of opportunities are there for pharmacy students in different fields. It can be regulations, it can be research and development, it can be quality control, it can be quality assurance, it can be hospital pharmacy, it can be community pharmacy, it can be clinical pharmacy, it can be academics, it can be setting up your own units or going to abroad for higher studies and job. future is very bright conditionally. The positive mindset with positive aim, if it is there, definitely success will be there. Sincere advice to all the members attending this, that try to visit all the official government websites of CDSCO, that is Central Drug Standard Control Organization, Government of India, NPPA, National Pharmaceutical Pricing Authority, IPC, Indian Pharmacopoeia Commission, Department of Pharmaceuticals, and Ministry of Health and Family Welfare, and try to see whatever the latest notifications and amendments are coming from time to time from these websites. They will be helpful to all the students and faculty for updating their knowledge. Remember, for updating your knowledge, there is no age, there is no time. So as and when you get time, it is time of social media. We can go on Google, we can go on Chat GPT, but remember, Chat GPT cannot be relied upon. This is only a basis just helping us. But otherwise, whatever the information you are getting from all these sources, try to use these sources for your review articles, for your research publications, and for your any article which you want to publish. With these remarks, my heartiest congratulations again and sincere thanks to Dr. Devinder Pathak and the entire team of Rajiv Academy for Pharmacy for giving me this opportunity to share my views in front of all the participating members. I am seeing 160 plus members attending this FDP program virtually. So my best wishes to each and everyone, and I hope that after the completion of 6 days of this FDP, everybody will be enriched in their knowledge, particularly in their specific field, what they want to choose. So my best wishes to all of you. And once again, thanks for inviting me to this webinar. Thank you very much.
Thank you so much, respected sir, for your valuable insights and motivating words. Now, without wasting as much of your time, I would like to invite our guest of honor, Professor Dr. Arun Garg, Vice Chancellor, MVN University, Palwal, to address the audience. Before addressing the audience, if I am talking about the profile of our honor, guest of honor, Professor Dr. Arun Garg, Vice Chancellor of MVN University and General Secretary of Indian Pharmacy Graduates Association, is a distinguished academician with over 37 years of dedicated service in pharmacy education, a gold medalist from Delhi University 1986 in pharmacology. He earned his doctorate from Tamil Nadu Dr. MGR Medical University under the mentorship of Dr. B. Suresh, President of PCI. His acclaimed research on hepatotoxicity and liver regeneration earned him the prestigious Dr. B. Mukherjee Award for the best paper in Indian Journal of Pharmacology, as well as Hamdard National Foundation Research Award for his work in Chinkara. Dr. Garg's excellence in teaching and research has been recognized by multiple honors, including the Young Teacher Award by SPER 2012, IPGA Professional Excellence Award 2013, IPGA Fellowship Award 2014, and the KC Chatterjee Memorial Award 2016. Over his illustrious career, he has significantly contributed to advancing pharmacy education and research through guiding numerous postgraduate and doctoral scholars while maintaining a strong commitment to innovation and quality in academics. So, I would like to invite Dr. Arun Garg sir. Please come, sir, and address the audience.
Good evening, all of you. Am I audible?
Yes, sir. Good evening, sir.
Good
Audible,
evening.
sir.
First of all, I am very thankful to Rajiv Academy of Pharmacy for inviting me as a guest of honor in this prestigious faculty development program. I am also thankful to Professor Devinder Pathak ji for conducting this program under the banner of IPGA Mathura local chapter. I am also very thankful to and feel proud that I am in Palwal and they are in Mathura, and all the faculty members who and all the students and other faculty members who have joined this program. I will start with a shlok of Gita, Karmanye Vadikaraste Mahaphaleshu Kadachana Mahakarmphal Heturvu Mate Samgosta Karmani. So the topic, in Gita, Lord Shri Krishna said that you keep on doing the work, you keep on doing good work, and the result of that will be provided to you by me or the Lord. Don't worry about the result. Ultimately, you will get success and you will feel proud of you. And exploring innovation in pharmaceutical sciences. It is really difficult to come up with a new drug, and there is a lot of research going on in herbal, in biotechnological products, in the area of anti-cancer, anti-diabetic, and several other fields. But to me, Indians are really very bright. Indians are hardworking, and India is doing great. India is rising in every field. You can see our Honorable Prime Minister leading the country in different avenues, and today we are not dependent on several things to the European and other countries. So keep on doing hard work. And the other thing which I want to tell you that most of the CEOs of the multinational companies are Indians. And we have the brains, we have different ideas. You young students and faculty members, you have different ideas. And as Dr. Nasa has told, that this is the age of AI. AI is doing really wonders. You can come up with the new ideas, new entrepreneurship, and you can develop a good professional career. Regarding pharmaceutical, there is no dearth of career avenues. Lot of career avenues are there, and you can choose any career and you can be successful in that. To me, for all pharmacists, the clinical research, pharmacovigilance, clinical studies, these are the few very potential areas where you require knowledge of your pharmaceuticals, or you can say pharmacology, and also communication skill and documentation skill. And then you can join any clinical science or any clinical research organization which can give you a lot of benefits and also a very bright future. But today, what I see personally is that the students are not doing the hard work. They want only the shortcut and they study only for just passing. But if you love this profession, if you love these subjects, then definitely you will love to cure the human. You will love to have attachment with the humans and the suffering or the diseases which we they are having. So love the profession, do the hard work, and then the success is there. So we are at Indian Pharmacy Graduate Association are trying hard to see that all the pharmacists wear the white apron or white lab coat, so that the prestige of the pharmacist is also improved. I urge all the professionals, all the faculty members, all the students who are going to the career of pharmacy or practice of pharmacy to wear this lab coat. National Pharmacy Week is also coming, so try to see that the pharmacist wear the lab coat, and the pharmacy is maintained with the high standards and ethics. Don't sell the narcotics and other drugs which are not, don't sell the antibiotics without prescription. These are some of the very general things which we should do and practice at ourselves. Tell the students not to give any antibiotics, and generally there is a lot of antimicrobial resistance we are seeing only in India, not in other parts of the countries. Okay, I once again thankful to all the members of the IPGA Mathura local chapter. I request all the participants who are attending this program to become the member of the Indian Pharmacy Graduate Association, which is one of the top very bright association at national level and is having around 10,000 members, life members, and is doing very well in all parts of the country. I also request the students to be the member of our student forum of IPGA and do several activities in association with the Central Student Forum. Thank you all. Jai Hind, Jai Bharat.
Thank you so much, sir, for your gracious presence and inspiring address. Thank you once again, sir. Moving ahead to the most awaited part of the evening, the expert session. We are privileged to have with us Dr. Vijay Sharma. Dr. Vijay Sharma is a distinguished academician and researcher currently serving as professor in the Department of Pharmaceutics at the SGT College of Pharmacy, as well as Dean Students Welfare at SGT University, Gurugram. With a PhD in pharmaceutical sciences and 19 years of dedicated experience, he has made significant contributions to teaching, mentoring, and research in pharmaceutical sciences. His expertise spans pre-formulation studies, novel dosage form development, targeted delivery systems, life sciences, clinical trials, and regulatory affairs. Professor Sharma has published over 60 research papers in esteemed journals, guided numerous research scholars, and earned multiple awards for innovation and academic leadership. Renowned for his commitment to educational advancement and holistic student development, he brings a progressive and impactful perspective to pharmaceutical education and research. So, without wasting as much of your time, I would like to invite Dr. Sharma to kindly take over the session, sir. Dr. Sharma will be enlightening us on the topic advancing pharmaceutical dosage forms through 3D printing technology. I request to Dr. Vijay Sharma to kindly take over the session. Please, sir.
Sir, please unmute yourself. Vijay sir, please unmute yourself.
Hello, Dr. Mayank. Am I audible now?
Yes, sir. Yes, sir. Yes, sir. Good
Okay.
evening, sir.
Very good evening.
Hmm.
So first of all, thank you, Dr. Pawan. And it's really my pleasure that my alma mater is organizing such a beautiful event that is none other than this e-FDP, and invited me to be the part of this particular FDP. I still remember whatsoever I am now is just because of my alma mater, and that is none other than Rajiv Academy for Pharmacy. Whatsoever we have learned, that is the pure grace of my teachers, my mentors, Professor Kamla Pathak, Professor Devendra Pathak, and all the faculty members at that time. I still have so many beautiful memories that Rajiv Academy possessed in me. So without wasting my time, I thank you, all the organizing team members of this FDP, for providing me this opportunity to join this FDP as an expert. Though I still consider myself as a student of pharmaceutics, because I believe that nobody in the world is complete, and the education is an ongoing process. जितना हम सीखते जाते हैं उतना हम expert बनते जाते हैं. So let's move ahead towards the topic. Dr. Mank, I think I am audible and my slides are
Yes, sir.
visible
Yes,
to
sir.
the
Yes,
screen.
sir. You are audible. You are audible, sir.
Okay, that's great.
Ah.
So as a topic that has been provided to me is the advancing pharmaceutical dosage forms through 3D printing technology. We all know that in the modern era, there are so many technologies that are involved for development of new dosage forms with so many concepts that somewhere going to modify the release pattern of the drug. But there is something else that also needs to amplify, that is the personalization of the dosage form, the fixation of the dosage form, and moreover, the tailoring of the dose and the combination therapies that are readily implemented these days. So considering all these challenges, the thing that is mainly should be into the practice, that is the combination that needs to be maintained without tampering, or we can say that without posting the dosage forms, is none other than the newly developed technologies which comes under the broad arena of 3D printing technology. I don't know why I'm facing the challenge in changing my slides.
Sir, any problem?
प्लीज एक्सक्यूज मी, बहुत ज्यादा बारिश हो रही है यहाँ पर और अभी अभी बिजली गिरी है बिल्डिंग के बाहर. I think there is some technical error. Please give me a couple of minutes. I'll fix it down.
No problem, sir. No problem. We will wait.
Hello, Mr. Pavan.
Yes, sir. Yes, sir.
I hope you can take the full slide in your email and you can display so that sir can go for discussion.
Okay, sir.
You
Okay,
can,
sir. No problem.
I think so that will be
Kindly,
better
kindly
because
mail me.
yeah, you just tell to sir to mail your full PPT so that you can go for presentation. He can give the explanation.
Sir, Mr. Samidhi Mukherjee will contact you soon. And Mr. Ritik Verma, she is technical operator. She will contact you soon. He will contact you soon, sir.
Mr. Pavan,
Yes, sir.
आपको मेरी स्लाइड्स मूव होती दिखाई दे रही हैं अभी?
No, sir. No, sir. Mr. Samidhi Mukherjee will contact you
सर
soon. Now,
मूव हो
now
रही
it
है
is
सर.
correct, sir.
Now it's moving now?
हाँ,
Yes,
yes
yes,
sir.
yes.
Yes
Okay,
sir.
okay. Thank you. Okay, so let me continue. Sorry for the delay.
ऑन कैमरा तो ये यूट्यूब पे लाइव देख रहे हैं सब.
Thank you for suggesting me कि मुझे YouTube पे सभी लोग लाइव देख रहे हैं. So shall I continue?
Yes sir.
Okay. So the challenges that raised in the pharmaceutical manufacturing is nothing else that I already quoted, that the fixed formulations are the major challenges. And the fixed formulation whatsoever is available on the market is nothing else but undoubtedly it is the combination of so many medicines in a single dosage form, or they have to be developed in a single dosage form. But the problem is that every patient needs a different combination with this different concentration. So the development of the fixed formulation is undoubtedly the basic challenge. And the growing demand as per the need of the patient is undoubtedly is a major challenge for any pharmaceutical manufacturer. So introducing the three-dimensional printing, that is also called as various technologies that comes under 3D printing technology, is a technique of creating 3D items from digital models by fusing or depositing material on a successive layer. It is nothing a particular innovation, but we can imagine that a particular surface is available for casting a new structure. And that casting of new structure is involved the dispersion of the raw material that we called excipients in our term, and the dispersion of the drug within that particular system in any particular geometry that we are basically visioned for. किसी भी geometry में, किसी भी shape में, किसी भी direction में, we can cast a structure, and that structure may lead to the development of a very beautiful and innovative dosage form. And that casting of the structure is done by the layer by layer deposition method. This particular complicated technology got the approval by FDA in August 2015. The 3D printing is nothing else but it is a total game changer in the arena of dosage form design. As you can see in the slide, there are so many different dosage forms available with the different shapes, different size, and different imprinting on their surface. On a very top diagram, you can see that the dosage forms are available in the Braille language. That if any particular patient who is having the compromised vision or with the ill vision, they can also identify their own dosage forms to be taken, to be administered to any particular patient easily. And this technology is only, or this technique is only feasible with the 3D printing technology. There are few more pictures which they basically reflecting the innovation or the innovative dosage form that have been developed by the 3D printing technology. Any shape, any size, perforation, so many channels to release the drug successfully is available only when such type of dosage forms have been developed using 3D printing technology. Now, how this 3D printing workflow works in pharmaceutical industry? The very first thing is that this is as same as when we cast any particular physical structure. It is not that easy that we blend the mixture, blend the drug with the excipients, and just going to compress it and the dosage form is ready to deliver. It is not that easy. It is basically the totally engineered vision to the development of a dosage form that is the arena of the pharmaceutical scientist. So the very first thing is that we need to cast a design, and that design is possible by the CAD. We all have heard about the CAD designer. So the CAD is basically used to design the internal and external structure, that is basically the geometry of that particular structure, which can be designed prior to the development of dosage form in a computer system. And once the CAD design has been completed, the very first thing that comes in mind that what needs to be selected to cast that particular structure, and that is nothing else but it is the material. Undoubtedly, we need to feed all the material that is excipient along with the API for casting or for the fabrication of the entire structure, that is 3D printed structure. To move forward, it is the printing process which takes place with the use of so many type of printers. These are 3D printers which are available commercially in the market, as well as which are available in the labs as well. And post-processing, they need to dry, they need to cure, they need to coat, and followed by the final step that is called as quality testing in the pharmaceutical industry. So moving ahead, let's move towards the various type of printers and printing technology which are available in the labs, and they are used for the printing of dosage forms. The very first technique is called as thermal inkjet printing. We can see the diagram is available, which is mainly composed of the silicon substrate and the photo-imaginable polymer. That photo-imaginable polymer is basically the ingredient that needs to cast a particular structure that is called as dosage form. that when get it melted with the high temperature, it just form an ink droplet. In form of inkjet printer, it is called as ink, but originally it is the melted form of the polymer, which when poured on a particular surface, it take a shape of a particular structure which we have already designed or fed into the CAD system. So this is basically the working of the thermal inkjet printing system. Now on to the other side, the inkjet printing is a versatile and non-contact deposition. It is a very beautiful and a hygienic technique that found the precision-based dosage form, which mainly involves the deposition of ink or the functional material drop by drop onto various surfaces. And this is basically the casting of the ink without using a paper onto a surface that is basically acted as a base to create a new structure, and that structure is nothing else but it is the dosage form which is 3D printed. Here we can see the inkjet printing, which is basically used the support material and the build material. Here the two type of inlets are providing. One is providing the support material, another one is providing the build material. And when the mixture of these two, the combination of these two dropped onto the surface, they act as a structural development onto the print heads. The milling head is working simultaneously, and the particle collectors collect the drug onto the surface, and it creates the overall structure development that can be seen easily in the drive. The next technology, which is the most working technology, and this technology is widely used these days, that is called as fuse deposition model. The technology is widely available these days in so many advanced laboratories. Undoubtedly, where I am working in my lab, this technology that is called as fuse deposition modeling is also available, and we have casted so many dosage forms successfully. So this fuse deposition modeling offers simply called as FDM and is available with so many features that it functions by melting a thermoplastic filament. When we are using the term filament, it is nothing else, but it is also the filament type structure which is made up of the polymer. And these polymers are controlled, and these polymers are nothing else, but they are the same polymers which have been prefabricated in terms of filament, and they have been fed in the fuse deposition modeling print type of printer. These printers are readily available in schools, universities, and so many other laboratories for the working and for the experimentation purpose of 3D printing based development of the dosage. The next technology is undoubtedly made a very, very beautiful change in the development of the pharmaceutical dosage forms by using 3D printing technology, because this technology has made a eureka in terms of 3D printing, while it has been considered and it has been approved for development of 3D dosage forms. Because this technology that is called as zip dose technology was the very firstly accepted technology for the patenting system, because it has developed the very first marketed formulation that is been available in the next slide. So the zip dose represent the paradigm shift in the pharmaceutical manufacturing, moving towards a future of personalized on-demand medication that is being tailored to individual patients' needs. Yes, the dosage form that I have talking about was Spritem, that was the very first FDA approved 3D printed pills, which was which was developed by using zip dose technology and was developed by Aprecia Pharmaceutical. It was nothing else, but it was a rapidly disintegrating tablets, and it was just a sip of liquid due to porous structure. It will form a porous structure, and these are the swallowing pills for the children's and elderly. The next technology is hot melt extrusion. Undoubtedly, we all have gone through the hot melt extrusion technology. It was used for so many things. It was these days it was used for granulation. These days it was used for preparing stencils. These days it was used for microspheres and so on. So this hot melt extrusion technology is now these basically collaborated with 3D printing technology, and it is widely used for the development of new drug delivery systems. So this hot melt extrusion technology is a robust processing technique that involves melting polymers and active pharmaceutical ingredients together. Then they are being forced to melt and the die to create the continuous form. These forms can range from thin films, small pellets, to even complex medical implants. When combined with the advanced 3D printing techniques, this technique allows for the predecessor control over drug release profiles. This means it is capable of creating medication that releases their active ingredients at specific rate or time. This technique is widely recognized and utilized across both the pharmaceutical industry for enhanced drug bioavailability and taste masking as well. In general processing, for developing new plastic products with superior properties, it's a versatile and scalable manufacturing method. SLA, that is also called as stereolithography. It is basically the light cured precision printing. Light cured precision printing means the UV laser technique is involved in such type of printing, and the development of dosage form is very much easily and secure, as well as the precise dosage form are being delivered. SLA is the 3D printing technology that uses a UV laser to selectively cure or solidify liquid photopolymer resin. The laser tracks the cross-sectional area by using 3D model, layer by layer. Starting from the first slide till now, we have used this layer by layer very frequently. This layer by layer means whenever we are going to cast any particular structure that we have already fed into the CAD system. जब हमने computer system में एक बार CAD के structure को feed कर दिया, तो all the dosage forms are not automatically developed at a particular point. They are being developed layer by layer. पहले एक लेयर कास्ट होगा, फिर दूसरा लेयर कास्ट होगा, और फिर आगे के प्रोसीडिंग लेयर्स जो है वो ऑटोमेटिकली कास्ट होते चले जाएंगे, एंड द लास्ट लेयर इज गोइंग टू बी completed and set to be the fully developed or resulted in the fully developed dosage form by using 3D printing technology. The resolution and smoothness that SLA produces is the primary advantage of its ability to produce incredibly high resolution parts with exceptionally smooth surface finishing. This is the beauty of 3D printing, and especially the SLA technique, that the finishing that this particular technique possesses and the resulted is unmatchable with the other techniques. This makes it ideal for application where fine details and aesthetic quality are paramount, such as intricate prototypes and consumer-facing products. There are so many diverse applications based on these unique features of SLA. SLA is extensively used in industries requiring precision and customizability. This includes creating highly accurate dental models and aligners, intricate jewelry models, and specialized medical devices component with biocompatible resin. Now the next technique is called as selective laser sintering, that is also called as the SLS technique. That is nothing else but it is the powder bed fusion for durable parts. The SLS technique is a powerful additive manufacturing technique that employs a high powder laser to selectively fuse powder material. Let's imagine a powder is available and that powder is to be casted as a structure, and that structure is a very rigid structure that has been resulted by the 3D printing technology called as selective laser sintering. A thin layer of powder is spread over the built platform, and the laser scan, the cross-sectional part is heating the powder to just below its melting point, causing the powder to sinter or fuse together. इतना ज़्यादा heat apply करना है कि that powder get melts and it get fused together. This process is repeated layer by layer again until the entire 3D object is formed within the powder bed. Now, the one important thing that not to be neglected, whenever we are going to apply the heat to this particular powder bed, they have to be applied within the cast. That is means within the mold. Cause the desirable structure that we are going to fabricate should be within the mold, or that should be the replica of that particular mold where the powder has been filled and the heat has been applied. Whenever the powder gets fused, it should take the exact structure whatever the mold is possessing. The complex geometry is eliminating the need of additional support structure. So the SLS is highly valued for producing small, strong, functional and durable parts suitable for end-use application. Now there are two techniques which are somewhere identical to each other, that is stencil printing and digital light processing. That is why I have created a similar slide for this. Stencil printing, as the name indicated, that we need to create a mold that is also can also be called as a stencil. Here you can see that a blue colored stencil, that is a blue color roller, which is basically containing or consists of the molds that are called as the stencils, and the powder bed has been blown through it. The entire powder is get the same structure and resulted into the final dosage form whenever this particular roller get rolled onto the conveyor belt. It is following the process of drying as well as analysis for the any type of imperfections, and finally the product has been casted. So the stencil printing is the foundational technique, especially in electronics manufacturing, which involves applying the paste such as solder paste or inks through a precisely patterned mask into a substrate. This method ensures consistent and accurate deposition of the material. And apart from this particular electronic manufacturing, this particular technique is nowadays is also implemented in terms of pharmaceutical development. The next technique is known as digital light processing. The digital light processing is another powerful additive manufacturing technology which is similar to SLA, but with a key difference that instead of a single laser point, it uses a digital simultaneously, and this flash curing makes DLP a significant faster. Undoubtedly, in the other techniques, we need the trying to get the created object dry to analysis it, but this technique is somewhere very unique by providing a very perfect structure of a desirable product. Now, the another technique is laminated object manufacturing. It is the layered sheet bonding. As we have already discussed that this 3D printing technology involves the casting of the structure layer by layer. But the bonding between the layers is the most important phenomena which remains or which led to the structure intact. So the laminated object manufacturing is an additive manufacturing process that builds 3D object by bonding and cutting successive layers of sheet material. These material can include paper, plastic, or even metal folds, which are typically coated with an adhesive on one side. The process involves a heated roller that presses down on a fresh layer of material, bonding it to the previous layer. This laser or another laser or knife then cut that particular outline of the part of the specific layer, which basically used to remove the unwanted material, and the cross hatch to facilitate the removal later. This LOM technique is a cost-effective method for creating larger prototype and concept models, especially when they are using expensive material like paper, if it offers rapid fabrication of design verification and form testing. Though the finished product typically requires post-processing to remove the excess material and achieve the final finish, but still the laminated object manufacturing is somewhere used by the manufacturing industry to develop the 3D printed pharmaceutical dosage. Now, this was all about the 3D printing technologies or the type of printers which are commercially available and are into practice by so many researchers, maybe pilot plant labs, or maybe by the industry. Now let us move towards the early phase development revolutionized, which is basically involved so many things time by time or stages by stage. So it involves the rapid prototype. uh, we should go a bit earlier and just thought whenever we are thinking about to develop our new dosage form that is basically involved a very innovative technology which is not readily available in the market, which is not readily available in the industries as well. So development of a prototype is a very big challenge. But nowadays, 3D printing technology make it very feasible and easy to develop a rapid prototype in a very faster way to allow quick creation of small batches, adapting to shifting clinical needs and accelerating development of the cycles. It undoubtedly reduces the time to market because first of all, we need to think of the vision to develop a prototype, then the development of the prototype. If the prototype gets successful, we need to go for the pilot plant scale up. And after the pilot plant scale up, we need to go for several analysis and then the product may be ready for the market launch. But nowadays, whatsoever we think, whatsoever we have vision, whatsoever we have casted to think that is readily available to develop and to launch into the market. So the time has been readily reduced to launch it in the market. That is why the streamlined drug formulation optimization significantly cutting down to the time required to bring it to any new drug to the market is very much easy through using these 3D printing technology. Now the efficient testing. पहले टाइम पे हमें बहुत सारी चीजें देखनी है because somewhere it is not readily mixed, somewhere the blends are not that much perfect, somewhere the punching machines or the die cavings are not that much uniform. So fluctuation of the dose, fluctuation in the size, fluctuation in the pressure, so that results in the fluctuation of the tablet hardness or anything else are the major parameters that needs to be testing before passing of the batch. But now all these steps are being vanished off just because of the launch of this 3D printing technology, because it is readily available, it is so much precise, it is very much useful technique to develop a tailored dosage form so that the efficient technique, or this can be termed as a technique that can wipe off all the analytical parameters. Now, here comes the role of the pharmacy personnel and the future outlook, that what we need to implement, how we can carry forward this 3D printing technology into the healthcare system. Undoubtedly, since 2015 and now we are in 2015, so many development has been made. But the thing is that we need to think that are we here only to publish the papers? क्या हमारा सिर्फ और सिर्फ ऑब्जेक्टिव ये है कि कोई भी एक पर्टिकुलर अगर टेक्निक आती है तो उस टेक्निक के आने के बाद हम एक उस टेक्निक को एक्सप्लोर करें, एक्सप्लोर करने के बाद हम एक पेपर पब्लिश करें, एंड दैट इज इट द ओनली गोल ऑफ द फार्मास्यूटिक्स? नो, I don't think so. There are so many areas yet need to be explored. So any particular technique that has been developed by any particular scientist should have the clear vision that how far we can carry and how should it could be forwarded to the next generation to develop and to explore it as much as they can. So there are so many areas need to be developed, need to be explored, so that the decentralized manufacturing can be made possible. The pharmacies could become localized production centers manufactured personalized medicines on demand with the due approval of any particular regulatory body. In addition to that, the digital healthcare integration is readily possible, is widely possible, and yes, it is possible so that the seamless integration with the digital health platforms for patient-specific dosing and real-time adherence monitoring is made a new area, the new generation, and the successful dosage form that can cater the need of any individual patient. And apart from that, the point of care principle. By 2030, a vision of fully customizable multi-drug tablet printing directly will be available at point of care. Now, what should be the impact of this particular type of technologies towards the truly personalized medicine? Yes, there will be the enhanced patient outcome. These days, or since the inception of the dosage form development, we being the students of pharmaceutics always care for patient compliance, and patient compliance. But is the patient compliance only desired that the dosage form has to be delivered safely and it should show its therapeutic effect? Or the patient compliance also demands that the patient needs to need that particular dosage form that is solely made for him or her as per its need, as per its demand, or as per its condition. So by adhering such type of technologies that are called as 3D printing technologies, the enhanced patient outcomes can also be visioned. So it has the potential to significantly reduce the side effects, improve drug efficacy, and enhance overall patient compliance due to customized dosage. Now, apart from that, we were also thinking about the targeted drug delivery system. But yes, 3D printing made it very easy. It enables the practice treatment of rare diseases and pediatric patients who often require highly specific and tailored drug doses. I'll give you the example in the next few slides. Apart from that, the supply chain transformation, which could fundamentally reshape the global pharmaceutical supply chain, will shift towards the agile, on-demand local production at scale. So, this 3D printing technology is not only limited to the high-scale industries as well. The pilot plant industries can also think of it with the due approval of the approving authorities. They can also be the producer of the tailored dosage form by using 3D printing technology. Now let's move towards the application of 3D printing technology in the development of pharmaceuticals. As we have already discussed that it is the paradigm shift from mass production to personalized medicine. So the traditional produces the fixed doses for average patient, often leading to the suboptimal efficacy and adverse reaction. The 3D printing enables tailored formulation, which allows on-demand patient-specific drug formulations tailored by dose, shape, release profile, and beautifully the combination therapy. अब तक हमने बात की या अब तक हमने मार्केट में देखा था that the combination of 2 drugs, 3 drugs, even sometimes the 4 drugs is available. But incorporating those 4 drugs without any interaction, without any side effect, and the perfect release pattern, which is being predetermined before casting that drug delivery system, is very difficult. But now 3D printing technology made it very possible and very easy that as we are predetermining that कि इस पर्टिकुलर ड्रग का रिलीज इस तरीके से इस टाइम पर इस पीएच पर होना चाहिए, it beautifully made it possible that yes, traditional dosage forms कहीं ना कहीं फेल हो सकते थे, but 3D printing technology made it very easy and possible that all the predetermined assumptions are going very smoothly in a same way, in a same fashion that we have casted in a CAD structure. So addressing the patient non-response, up to 70% of patients do not respond well to convenient drugs, while 3D printing offers a solution to improve therapeutic outcomes and adherence. And this is a very big success of 3D printing technology. Now, when it comes to diverse pharmaceutical application, which is enabled by 3D printing, it is none other than the immediate and controlled release dosage form. Any type of dosage form can be developed. It is not the immediate one that can only be developed. It is not the control of the chain release that can be developed, but any type of dosage form followed following any type of modified drug release systems can be developed by using this technology. Microneedle patches, yes, a very beautiful thing. Till now, whatsoever is possible, that we have casted so many type of transdermal patches which may contains the involvement of the electronics, which may involve the electronic of ion exchange, which may involve the passing of the drug by its own partition coefficient. But nowadays, the microneedle patches can easily be developed by using the very fine needles of 3D printers, and they are successfully working. Polyfill, yes, the another success story that is based on the 3D printing, that is developed by a researcher and is available for our reference. Let's see the concept of the polyfill. Here we can see that there are this particular dosage form, that is the polyfill, is the combination of 5 drugs in a same dosage form. Here we can see that there are 2 different layers. The very first layer is of immediate release compartment, and the another, that is the broader layer or the lower layer, is the combination of 3 drugs which is for extended release. The 2 drugs which is available in the immediate release compartment is nothing else but it is the combination of hydrochlorothiazide and atenolol, while the lower drug composed of ramipril, atenolol, and pravastatin. This is the dosage form that has been developed by Khalid et al. And is being termed as polyfill. Poly, the multiple drugs, poly representing the multiple drugs, and they all are composed. They all are composing a single dosage form, that is nothing else but it is a pill, and they are available in 5 different chambers. Lower layer is made up of 3 chambers, while the upper layer is the single layer which composed of 2 drugs. So 5 drugs with different type of release pattern in a same tablet in a same pill. And the dissolution data shows that more than 75% of the aspirin and hydrochlorothiazide were released within the first 30 minutes, which is the desirable criteria of any type of immediate release dosage form. While the second layer, that was the extended release compartments, release the drug over the period of 720 minutes as it requires with 69, 81, and 66% drug release respectively. So this made another success story that has been made possible by Khalid et al. And it is nothing else but it is the polyfills. Now, the question arises that as of traditional method, is there any regulatory challenges or any particular landscape that needs to be followed for the development or for analysis of such type of dosage forms that are made possible by using 3D printing? But yes, there are technical hurdles during material selection and printer precision. They still possess challenge. As we gone through the pre-formulation aspects, we always select a suitable material which is non-reactive, which is non-interactive, which made the dosage form possible to release its drug very easily. So the same is the point which is also faced by 3D printing technology. The regulatory evaluation, which is the framework which is evolving to address quality, safety, and reproducibility of the 3D printing drugs. The ethical concern governs. It includes patient data, privacy, and potential misuse of printing technology. And the collaboration is the most desirable thing, which basically governs the critical partnership which is needed between the pharma companies, regulators, healthcare, and more importantly, with the educators and the research scientists which are available in academia. Now, the role of healthcare professionals in adoption. So, very big challenge nowadays is that are we acceptable to the new techniques that are available or being generated by the new scientists? We should. The pharmacists and the clinicians are the key to integrating 3D printing into clinical practice. It enables on-site hospital-based production of personalized medicines that is being tailored to patient physiology. The education and training are vital to support widespread adoption and ensure patient safety. So this 3D printing technology should be widely adopted, should be widely accepted, and it should come in the practice. Now let us talk about the future horizons. And the future horizons are bioprinting and beyond. I feel myself very lucky that I am currently working in such type of campus, which is having variety of courses and variety of schools within the same university. As we are having so many medical courses, we are having engineering in so many streams, we are having non-medical courses as well. And since we are available with such type of 3D printers in our university, I can easily see that so many dentures have been developed by dental scientists, so many structures have been developed by mechanical scientists, and so many dosage forms are also being developed by using 3D printing technology in our university. So the bioprinting and beyond that particular thing, we can think of developing any particular type of structure, any particular type of dosage form, any particular type of biomaterial that can be termed as the implant, that can be termed as a particular type of structure that can be delivered to the patient or that can be fitted or implant to the patient, so that the role of 3D printing technology can be successful, can be made successful by using all these techniques. So the future horizons may be in bioprinting for research, the regenerative medicines, the point of care printing that we have already discussed, and precision medicine integration. Because this precision medicine integration is the widely desirable point, widely desirable thing at this particular time, which is seamless integration with digital health initiatives to revolutionize treatment paradigm and personalized. Now, why 3D printing is critical for modern pharma? Yes, the personalized medicine, undoubtedly we are thinking of, we are in need of personalized medicine, but the vulnerable population overcomes the challenging in formulation the drugs of pediatric and geriatric patients with precise dosage. The complex therapies which made it possible, but somewhere they are also the result of failure. So the agile development that accelerate prototyping and reduce the time and cost associated with the traditional batch manufacturing can be made possible by using 3D printing technology. Now, expanding the horizon beyond traditional dosage form, we can also think of implants and scaffolds. And undoubtedly, there are so many scientists who have reported so many type of implants that have been developed by using 3D printing technology, and scaffolds have been successfully developed, tested, and got patented successfully. So the creation of devices for long-term or localized drug delivery in oncology and regenerative medicine made possible. Bioprinting integration, we have already discussed, and undoubtedly the smart drug delivery system, which is nothing else but it is the development of responsive system that release the drug based on biological triggers like pH or temperature. As we have already discussed, that there should be the regulatory landscape as of traditional method of manufacturing of the drug. So the regulatory focus is on reproducibility of the printing process, robust quality control measures throughout the manufacturing, product stability under various conditions, and adaptation of good manufacturing practices for 3D printing as well. Because these days, only 3D printing is being used to develop the structure, but they are, I don't think so that they are following the entire guidelines of the good manufacturing practices. So such type of things needs to be considered, and a complete regulatory focus should be on them. Apart from that, the quality control measure should be there by using comprehensive mechanical stress testing, precise resolution and disintegrating profile, rigorous stability studies against environmental factors, and verification of dose uniformity across all printed units. Undoubtedly, as of traditional method, there might be some of the challenges because till now the material, the filaments, the polymers that are very much acceptable by the available 3D printers are very much less. So there is a need to develop, to identify, to search more and more polymers so that we can use variety of polymers for development of variety of dosage forms as per the need, as per the predetermined rate of drug release. It may be immediate release, it may be sustained, it may be controlled, it may be pulsatile, whatsoever we wish to develop, we may develop by using the variety of polymers. So there is currently it is a limitation of material because very few polymers are very much friendly to these available 3D printers, but there is a need to identify so many new polymers, so many new filaments to be made to be incorporated into the 3D printers. Scale up hurdles, yes. One thing that is the basic challenge, the speed. In a speed that we can follow, we can develop the traditional dosage forms, the speed of the 3D printing is very much less. So we need to scale up, we need to make it possible, especially mechanical engineers can be requested or should think of that how such type of 3D printers are available so that the speedily process or the speedy manufacturing can be taken care of. The cost and validation. Undoubtedly, one side we are focusing on to the tailoring of the dosage form, we are satisfying the need of the patient, but simultaneously the cost and the validation of the dosage form is still high. So we need to think of that how the cost can be minimized, how the cost effective dosage form can also be developed by using 3D technology. It is the role of the working pharmacist nowadays. The regulatory navigation. So the overcoming regulatory approvals, hurdles, and achieving global harmonization for these novel products should be. Yes, the very beautiful line that the 3D printing is not just an innovation, it is a transformation. It definitely transforms the way of traditional development of the dosage form, the way we thought of that yes, in such a way the dosage can be tailored, the dosage of multiple drugs can be incorporated into the same system made possible only by 3D printing technology in such a beautiful and in such a successful manner. So redefining the drug development which enables rapid prototyping, efficient screening of the formulation and on-demand clinical trial batch. Apart from that, the personalized and precise dosage forms are being developed, which shift the focus towards patient-centric designs, allowing for tailored dosage and complex multiple therapies. In addition to that, undoubtedly there are challenges of few innovations. So despite regulatory and scaling hurdles, continuous advancements are propelling the field forward. And the future inspect poised to improve global access to medicine, enhance supply chain resilience, and promote sustainable pharmaceutical manufacturing. So with these words, I thank you all for joining this particular session. I welcome all the questions if any particular audience is having. It may be asked now or they may be send it through the mail either to the organizing team member or directly to me at drvijay.pharmacy@sdtuniversity.org.
Thank you so much, sir. Now it's your turn to ask questions. Please feel free to ask your question, Dr. Vijay Sharma, sir, about advancing pharmaceutical dosage forms through 3D printing technology. Who has a question? Sir, I have received a question from Lalit's side. Why we use UV radiation in 3D printing?
See, this UV radiation is not only to guide or to make that 3D structure possible, but it is also to such an extent so that the heat is developed for the CAD structured design. The UV radiation is the only possible thing that can make the 3D casted or the CAD casted design in such a geometry, such perfect geometry, so that an entire structure can be framed easily. Such rays basically guide the filament or the melted excipient to that particular direction so that each and every angle of the casted 3D design has been made possible and the perfect shape has been obtained.
Thank you so much, sir. And that's all the time we have for today's discussion on advancing pharmaceutical dosage forms through 3D printing technology. We have explored various techniques, including thermal inkjet printing, fused deposition modeling, gyptos, and laminated object manufacturing. I would like to thank you, our esteemed speaker, Dr. Vijay Sharma sir, for shedding light on these innovative technologies and their potential applications in pharmaceuticals. His insights have given us a deeper understanding of benefits and possibilities of 3D printing in creating personalized and complex dosage forms. Thank you, Dr. Sharma, for an enlightening presentation.
Thank
Thank
you,
you,
Mr.
sir.
Pawan Pandey, and thanks, Mayan.
Thank you,
Sir,
sir. Thank you so much. Thank you, sir.
thank you so
Thanks
much.
to the organizing team of this FDP for providing me such opportunity, and thanks to my alma mater, Rajiv Academy for Pharmacy.
Sir, we have some another questions from audience side. Question is raised from Kamlesh Patel. Which UV radiation is used for bioprinting, UVA or UVB?
See, there is the selection of the UV radiation is totally depend on the material that is to be used. We are using these days, initially it was UVB, but these days UVA and UVB both are being used. That is totally depend on the selection of the type of material that we are using as an excipient. So the type of polymer that we are using is the total guidance or the acceptance of the type of UV rays that we are using for 3D printing material.
Okay, sir, thank you. Sir, another question. What are the limitations of 3D printing?
What there are so many limitations that we have already discussed, though I didn't put it the heading of limitations, but we have already discussed in during the discussion. The very first thing is that the availability of the very limited polymers that are 3D printers friendly. Number two is the very slow speed of development or the production. And yes, Mr. Nitish Bhatia, that he is asking for is there the possibility of colon drug delivery development of the colon drug delivery system by using 3D printing? Sir, I wish to tell you that yes, we and our team has successfully developed the colon targeted drug delivery system by using 3D printing technology. Though I didn't present it, or I didn't accommodated it in my slides because we are that particular research is still under process, and we are still yet and we are still yet to publish it. So let me publish it, and I will definitely going to share it with you that how 3D printing can be utilized for colon targeted drug delivery.
Okay. Sir, question is from Parvesh Kumar. Mr. Soumyadeep, sir, kindly send me question. Okay, okay.
I think all have done. I think.
Question is, what is the ideal criteria for selection of APIs in polyfills formulations?
Okay. The ideal criteria is, as we have already gone through various type of technologies of 3D printing, the ideal criteria is the drug should be stable at a very high temperature. The very first thing, because there are any particular technique that we are using these days for development of 3D printed based dosage forms, they all are based on or the processed with a very high temperature. So the very first thing that it should not be readily degraded on the application of temperature. The very first criteria. Another thing is that the melted form should be available or should be dispersed easily within the matrix system, because there are so many drugs which when get melted, they easily either get crystallized or they get degraded or they get charged. So such type of drugs should not be considered, and they are not at all the ideal candidate for development of such type of dosage form by using 3D.
Thank you so much, sir. Any other question?
Participants are free to open their cameras and mic to ask questions.
Thank you, Mr.
Participants,
Sachin
you can
Chaudhary.
ask directly.
One more declaration on behalf of all the organizing committee I wish to make that in my lab there are so many advanced techniques are available. SGT University offers various institutions and various research to collaborate. We would love to collaborate with any researcher, any student, any PG or PhD scholar to collaborate. If you wish to develop any particular dosage form by using 3D printing technology, we are open to collaborate with you. We, SGT University, welcomes you all to collaborate. In any case, if you feel like this, if you are having any particular concept, you can discuss me personally, or you can write me a mail, and my mail is drvijay.pharmacy@sgtuniversity.org.
Hello.
Any other question?
Hello.
Yes,
Yes,
please.
Dr. Atul.
How are you? I hope so. This was a nice session. I just have a because I am also working in parallel with this all 3D printing tools. So for me, when you use Ultimaker kind of system, because they normally use a thermoplastic ones, they're not compatible with biocompatible and FDA approved polymers normally.
Yes.
Is it so? How you think that we can use this Ultimaker printers when we are looking for any drug formulations, specifically when materials are not approved, like they are not compatible?
The thing is that it is not the question that materials are not approved. The major problem is that materials are not identified. These days, a lot of materials are available. If even I'm talking of my own self, I'm also available with very few type of materials which are biocompatible or which are friendly to the 3D printers which are available commercially or in the labs. So currently there is a need to identify more and more friendly materials that may be biocompatible, that may be 3D printer friendly. But yes, undoubtedly, we need to talk of so many mechanical engineers as well, that they need to develop such type of printers as well, so that they can be friendly with the material which is available in the market or they are in practice of the research.
Yeah, thank you, sir. But what do you think about? I'm just. This is not a question. I think it's just for the discussion. What do you think about using hydrogel and electrogels using these printers?
See, hydrogels can be utilized, but the major problem that I have already mentioned is the problem of churn. If any material is not available or it is not stable at high temperature, we can't use at all in 3D printing technology. So hydrogel is, I think that they may be more challenging in comparison to the polymer. General polymer, hydrogels cannot be used. As of my knowledge, the hydrogels cannot be used by the readily available techniques, because they are mainly tend to apply a very high temperature for the melting of the polymers which are available now.
Thank you, sir.
Any other questions? Okay, moving ahead. Thank you, sir, for such an enlightening and futuristic session. I'm sure our participants have gained great knowledge from your expertise. As we come to close the day one, on the behalf of organizers, Professor Dr. Devendra Pathak, President, Indian Pharmacy Graduate Association, and Professor Dr. Himanshu Chopra, Director, Rajiv Academy for Pharmacy, Mathura, and Vice President, IPGA, Mathura Local Chapter, extend grateful gratitude to all our dignitaries, speakers, and participants for making this inaugural day a great success. Thank you, Dr. Mayank Kulsresh, sir, Organizing Secretary. We look forward to your continued participation in the upcoming days of this FDP. Thank you, everyone, and have a pleasant evening ahead. Thank you so much.
Thank you,
Thank
Vijay
you
sir.
all.
Thank you
Sir,
so much.
again, thank you so much.
Shall
Thank
I
you.
end the session?
Yes, sir.
Thank you.